Hi everyone, my name is Hazel Steven. I'm from Group 4 and I'll be presenting about research presentation, protein biomarkers as molecular tools for forensic biofluid identification. And these are the group members in Group 4, which is Nick Wadina Safia, Heidi Yang Chimbul, Nur Safia Wardini, and myself, Hazel Steven. Now we all know that DNA profiling is the absolute gold standard for human identification, but in the real world, DNA has major limits. It can easily fail if a sample is badly degraded by radon, if there isn't enough sample left, or if the scene is contaminated. On top of that, DNA alone can't tell us what kind of body fluid we are looking at, or even when it was left. That is where forensic proteomics comes in. By studying proteins instead of DNA, it gives us an essential backup safeguard when traditional genetic testing just doesn't work. So, what makes proteins are so unique and powerful for forensic science? First is their stability. Proteins are physically much tougher than DNA and can last a lot longer in highly decomposed or limited trace samples. Because of this durability, protein biomarkers can uniquely reveal things DNA cannot. For example, they can confirm the exact biological origin of a stain, telling us if it's blood, saliva, or semen. They track specific protein degradation patterns to help us calculate the post-mortem interval, which means estimating the time since death. They can also reliably separate human traces from animal traces in complex casework. Lastly, we can now even look at genetically variant peptides or GBPs. While they don't replace DNA completely, they give us a reliable alternative way to identify a person when the DNA is completely gone. To keep it simple, current forensic investigation face some main problems that our technology solves. First, DNA degrades easily. If a crime scene is exposed to a harsh weather, or if the sample is too small, traditional DNA testing fails. Even when we do get DNA, it can't tell us what kind of body fluid it came or exactly when the crime happened. Second, the old chemical tests we currently use are unreliable. They easily give false positive if the scene is dirty, and they rely too much on the analyst's visual guesswork. Finally, nature works against us. Microbes rapidly destroy evidence after death, and rough surfaces like denim can hide stains completely. Proteomics solves all of this by focusing on tough, stable proteins that survive these harsh conditions.
Okay, now I will discuss the analysis of biofluid biomarkers in forensic proteomics. Based on Table 1 in the article, different body fluids contain specific protein biomarkers that can be used for identification. For example, blood can be identified through hemoglobin, albumin, and hemopexin. Saliva contains proteins such as staterin and histatin 1. Semen is identified using seminoglin 1, seminoglin 2, and PSA. Meanwhile, urine can be identified through uromodulin and osteopontin. These biomarkers are very useful because they remain detectable even when samples are degraded. This improves forensic analysis, especially in crime scene where DNA evidence is damaged or insufficient. Next, Figure 3 provides a visual comparison of protein biomarkers across different biofluids. The bubble size represents peptide count, where a larger bubble indicates stronger peptide evidence and higher confidence of detection. From the figure, seminoglin 2 shows strong detection for semen identification. Staterin is highly specific for saliva, while hemoglobin is a reliable marker for blood. This is very important because it allows forensic scientists to distinguish between body fluids more accurately, even when samples are mixed or only present in trace amounts. Therefore, proteomic analysis provides stronger evidence interpretation compared with traditional testing methods. For this section, I will explain the alternative methods and solution options used in forensic science. Traditional forensic methods mainly rely on chemical and enzymatic tests. However, these methods have several limitations such as lower specificity, false positive result, and difficulty analyzing degraded or mixed samples. As an alternative, forensic scientists now use proteomic analysis through LCMS or MC technology. This method focuses on identifying specific protein biomarkers found in body fluids. Compared to traditional methods, proteomics provides higher accuracy and sensitivity. It can also work effectively even when the evidence is degraded or contaminated. Therefore, proteomic analysis offers a more reliable approach for forensic investigations. This slide explains how modern forensic science combines LCMS with artificial intelligence and bioinformatics. Machine learning helps improve detection accuracy and reduce human interpretation errors during analysis. Bioinformatics is also important because it helps scientists analyze complex protein data more efficiently. Some examples of important biomarkers are hemoglobin for blood, statin for saliva, and seminoglin 1 and seminoglin 2 for semen identification. These biomarkers allow forensic scientists to identify biological evidence more accurately, especially in cases where DNA evidence is insufficient or degraded. Overall, proteomics provides a more advanced, accurate, and reliable solution for modern forensic investigations.
Now, transitioning to the practical hurdles, the article highlights significant limitation. First, sample complexity. Unlike controlled lab environments, real-world crime scene evidence is often a complex cocktail of overlapping protein profiles from the victim, the suspect, and the contaminants. This data diversity creates a major transition barrier. Second, we face a problem of protein degradation. Environmental accelerators like fluctuating temperature and microbial growth trigger rapid autolysis, disrupting predictable decay patterns and undermining postmortem interval estimation. Furthermore, the strict requirement for immediate frozen preservation remain a constraint in technologically limited settings. Lastly, legal and ethical hurdles heavily impact judicial admissibility. It is because globally harmonized standard operating procedures are still lacking. Proteomic findings can be easily questioned by the legal defense teams. This gap highlights the urgent need for validated reference library to ensure that interlaboratory report.
Now, we will take a look at the recommendation and final action plan based on the article about forensic proteomics and protein biomarkers, which focuses on how forensic proteomics can be realistically implemented in actual forensic investigations, especially for body fluid identification and postmortem interval estimation. First, technological innovation. We can implement forensic proteomics in real casework by using liquid chromatography mass spectrometry as the primary analytical method. We can also combine proteomics with artificial intelligence, AI, and bioinformatic tools such as MaxQuant and malware to improve data interpretation and increase accuracy. This method provides a reliable alternative when DNA evidence is degraded or unavailable. Second, by using protein degradation marker to estimate post-mortem interval. Protein degradation marker can be used to detect actin, tropomyosin, and biglycan because according to the article, this protein will undergo degradation after death. This protein also can function as molecular clocks to estimate the time since death more accurately than traditional method. The article also mentioned that bone proteins and collagen can remain stable for long periods of times, making them useful even in highly decomposed remains. Third is protein biomarkers. We can implement this method for body fluid identifications. According to the article, liquid chromatography mass spectrometry have successfully identified lactoferrin in saliva, seminogenin in semen, and hemoglobin in blood, especially in degraded or mixed samples. These biomarkers are highly specific and can perform better than traditional chemical tests, which can sometimes produce false positive results due to environmental interference. Other than that, development of standardized protocols. The article highlighted that one of the biggest challenges is the lack of universal standards for sample extraction, mass spectrometry operation, and data interpretation. Therefore, laboratories should establish universal protocols and validated protein reference database to increase identification accuracy and accomplish effective forensic comparison. Next, forensic proteomics should be gradually integrated into routine forensic investigations through multidisciplinary collaboration. This includes cooperation between forensic scientists, analytical chemists, bioinformaticians, and legal experts. The article emphasized that future development requires standardized protocols, affordable portable technologies, expanded protein databases, and proper legal validation to ensure courtroom acceptance. Lastly, integration with other omics technologies. Complete knowledge about forensic evidence can be generated through the integration of proteomics with genomics, transcriptomics, and metabolomics via multi-omics approaches. This integration will result in stronger conclusions being drawn regarding tissue authentication, postmortem interval determination, and toxicological analysis.
While the practical protocols and biomarkers just mentioned give us an immediate solution for today's casework, the true evolutionary power of forensic proteomics lies in its long-term technological advancement. Let us now look at our first future horizon, which is smart framework through AI and automations. Smart frameworks will combine high-resolution mass spectrometry with a deep learning algorithm. This network will automate spectral interpretation, filter out microbiome noise, and eliminate human analysis. Next, the analytical toolkit is also expanding into toxicoproteomics. Future biomarkers will detect drug exposure and chemical poisoning through stable toxic protein adducts, even in degraded remains. Simultaneously, universal protocols will finally secure flawless courtroom acceptance. Lastly, the ultimate milestone is the transition into the foreign omics era. By seamlessly integrating proteomics with genomics, transcriptomics, and metabolomics, future forensic scientists can achieve a holistic 300-degree molecular reconstruction of any crime scenes.
In final conclusion, forensic proteomics serve as an indispensable molecular safeguard when traditional DNA profiling completely fails due to severe environmental degradation. It successfully shifts our focus from sole identification to comprehensive, context-driven crime scene insight. As automated frameworks mature, designs will fundamentally drive higher precision, absolute objectivity, and true equity in the modern criminal justice system. On behalf of our group, thank you very much for your attention.